Prompt 1. And so for prompt 1, we wanted to look at a protein that's 5 amino acids long and is formed from this DNA sequence in vivo. And so in this example, we see that the transcription is running from right to left. And so first we wanted to establish the coding and template strand. We know that in this cell, there's actually no left and right. So we had to figure out which would produce a 5 amino acid sequence. The bottom strand allowed us to do that. So that's our coding strand reading 5' to 3'. And the top strand is the template strand reading 3' to 5' direction. So to transcribe, we use the template strand in a 3' to 5' direction and add our perfect complement base pairs. That every time we see a C, we make it a G. Every time we see a T, we make it an A. Every time we see an A, we make it a U. And every time we see a G, we make it a C, etc. And so then we had to translate our mRNA sequence that we found. And so we wanted to look at the spot that had the first AUG because that's our start codon. And then from there, we wanted to look at all the base pairs in 3s to create a codon. And then now every codon is going to be used to create an amino acid, which can be found on this table. And so when we look at the codons that are color-coded, we see that we create MET, VAL, R, V, LIS. And so I wanted to think about this in terms of all of these are ingredients that are going to make some kind of lovely recipe. Like the amino acid is going to end up being, all the amino acids are going to end up being these awesome proteins that are going to create these changes in our cells. And so from the given DNA, we can make some kind, if we look at these as ingredients for baking, this is going to make some kind of awesome oatmeal cookie. But what happens if we have some kind of mutation that's going to alter the ingredients that are going to be added? So let's say we have a transversion where one of the amino acids goes from being a G to being a C. We're actually going to get a missense mutation that's going to alter our proteins. And so when we see it here, when we're trying to translate it, we see that we actually have LOU, which if we go back to the ingredients, coded for lettuce. And so instead of making oatmeal cookies, we're going to make this weird concoction with lettuce. And that's not really what we wanted to eat or our cells needed. So what if we have some kind of other mutation? One of the codons is stop codon. For example, we have a transversion from A to T, where we're going to end up having UAG and we're going to have a stop. So we could potentially still make oatmeal cookies, but they're not going to have the kosher salt that we need. So they might be like a little weird and not really function or taste the same, just like our proteins that are going to be made in the cell. We can have a silent mutation, which is actually no change. For example, if you have a transition from what used to be a G to an A, a code for the same thing if we go to our table. And so we'll still make the awesome cookies that we want. And then read through. Here we are making just ultimately too many things because we no longer have a stop codon. So let's say we had a transversion from G to C in this codon right here. We lose our stop codon and we're just going to keep adding things, make something that is not necessarily desired.